One glycerol, which would be a monomer, and three, each one of those three is a monomer, and join them together. So a fat is really a glycerol and three fatty acids. That's why I get the term tri. It refers to this. Glyceride refers to that. This is the beginning of a triglyceride. Here's your glycerol molecule, and you've just added a long chain of fatty acids. Are fatty acids organic?
No.
Yes.
Yes, because right, it's a carbon bound to a hydrogen. And this is actually called a hydrocarbon. That's all you've got are hydrogens and carbons, except for the tail end way over here. All right. Can I also refer to this as an intramolecular reaction? Is this intra or intermolecular?
Enter.
It's intermolecular because we're taking this fatty acid and joining it to this glycerol. Is this hydrolysis? Is this hydrolysis? Is this hydrolysis? Ladies and gentlemen, how many of you don't know? Raise your hands. All right, so what is hydrolysis? Right, lysis means to break, right? And hydro means water. So are we breaking this apart or are we sticking things together?
Sticking it
Are
together.
we synthesizing?
Yeah.
So we're doing dehydration synthesis, right? We're making something with this. All right, awesome. Then, when you're all done, just do it 2 more times. Another intermolecular reaction, another dehydration synthesis, another intermolecular reaction, more dehydration synthesis, and I have a glycerol and 1, 2, 3 fatty acids. What makes one lipid different from another is not this, but how long this is. This could be 20 carbons long, or 60 or 80 carbons long. That's what makes them different. And we know that because it's so doggone big, right, it doesn't dissolve very well in water. So what term can I use that says does not dissolve very well in water? It's hydrophobic, exactly. This is why science has a lot of terminology. You guys might take a whole paragraph to write out a description. I can do it maybe in 2 sentences, as long as I know the vocabulary, right? So now instead of saying, well, here's a substance that does not transport or dissolve very well in water, John, you simply say, oh, it's hydrophobic, right? It'll help you. Just practice them. All right, now, lipids we know are nonpolar. They're nonpolar covalent bonds. We know they do not form bonds with water because they are hydrophobic. Another kind of a lipid is called a steroid, or sterols. Steroids are hormones, like testosterone, cortisol. You ever had a cortisol injection, right? Cortisol is a steroid hormone. This is a monomer for steroids. This is called a cholesterol molecule. This is what cholesterol looks like in structure or form. Don't memorize it. I'll never ask you to draw it. But now let's go back and look at all these things. Now we can look at all of these large macromolecules that are of lipid forms. I've got a fatty acid here. That's all it is a fatty acid. Another different fatty acid. But now this one is saturated. This one is unsaturated. What does it mean to be saturated versus unsaturated? If every single carbon has all the possible number of hydrogens associated with it, I have saturated it with hydrogens. But there's a double bond right there. That means these two guys, there's no hydrogens here. So it is unsaturated. Now, fast containing long chains of carbon atoms bonded one to another. I can then call this a fatty acid. I can then join a fatty acid to a glycerol. If I do that 2 more times, I can create a triglyceride.
Okay.
Over here, you have what's called a phospholipid. Now, you do not need to memorize the structure of it. But what I want you to know what a phospholipid is it is a phosphate bound to a glycerol with 2 fatty acids. So this is a lipid right? With a phosphate, so phospholipids. That you do need to know, right? It is a lipid, so it's a glycerol with two fatty acids bound to a phosphate group. This, right here, is the monomer for making cell membranes. This is the building block of life in a sense. And we need to understand how that works. We'll get to that in a couple of days. Okay, now for a bigger one, a little more complicated perhaps, maybe a little more confusing than proteins for the moment. Now we're at nucleic acids. There are two types of nucleic acids. DNA is one. Anybody know the other one? RNA. That is the other type of nucleic acid. So you have DNA and you have RNA. And we're going to learn how you make both and what each of them do over the next several days. All right, so RNA and DNA are big polymers. That must mean they have monomers. All right, well, what is the monomer of nucleic acids? It's called a nucleotide. Whatever that is, they're made up of them. Okay, well, nucleotides are polymers. So that means they have to have their own monomers as well. That's how big and complicated nucleic acids are. They're polymers made of polymers, which are made of monomers. So what is the building block of a nucleotide? Well, it has three parts. It has a sugar, a phosphate, and something called a nitrogenous base, whatever that is. Okay, so these three things make up a nucleotide. And nucleotides make up DNA. This is where you want to layer the information, right? DNAs are made up of nucleotides. Got that? Nucleotides made up of these three things. Well, what kind of a sugar is actually in a DNA molecule? It is a D, and what does D mean? It means removal, right? Right? Oxy for oxygen, ribose for sugar. It's a sugar that has an oxygen taken off of it. So all you need to realize is that the molecule that we're using for sugar is called deoxyribose. That's where the D comes from in DNA. Deoxyribose nucleic acid. And we'll go through the more formal name a little bit later. That's a phosphate, right? That's a chemical element, and a nitrogenous base. Well, what is a nitrogenous base? Well, we've got 4 different kinds of nitrogenous bases. You have what's called adenine, thymine, guanine, and cytosine. You need to be able to spell them. If I were to ask you to name the nitrogenous bases, then you need to write the full name out. When you get really comfortable with adenine, thymine, guanine, and cytosine, like any of my fellow faculty members are, we really don't use those words anymore. We just talk about A's, T's, C's, and G's. Because we all know what an A is. It stands for adenine. We all know what a C is. It stands for cytosine. But you need to be able to tell me what those letters refer to. So I would always just go off of the actual name of the different types of nitrogenous base. All right, so here's where you stop, you pause, and you practice it again. All right, DNA, a sugar, a phosphate, and a nitrogenous base. All right, got that. Oh, what kind of sugar? Something called deoxyribose, whatever that is. All right. Now, how many different types of nitrogenous bases do we have? We only got 4. Adenine, thymine, guanine, and cytosine. Now, this is actually what tells your body how you work. This, in a sense, is your DNA. You've only got 4 letters we're using. But look how different each and every one of you are to your classmates. None of us are the same. And yet, evolution is, let's start off first over here. There's a phosphate. There's my sugar. There's my nitrogenous base. So this, I don't know if that's a sugar, a phosphate, nitrogenous base, sugar. Oh, that's a nucleotide. Okay, cool. There's one nucleotide right there. Now it's color-coded. Right, look at the yellow, blue, and green. Yellows and blues are only found over here. The greens come off to here. So a nice way of describing or thinking about the structure of DNA is a ladder. You have the sides of the ladder, which will be then our blues and yellows. The rungs of the ladder are nothing but the nitrogenous bases. Now take your ladder and twist it like a sugar cane or a spiral staircase. DNA is what's called a spiral or a helical molecule. Because now you're beginning to pack it up. You're beginning to make it smaller. When your cells use this, that's to unwind it, to make it more like a straight ladder, to gain access to the nitrogenous bases that are in here. So then, this is the beginning of your DNA. This is a sequence of an A, a T, a C, a G, and a T. And it goes on and on and on and on for maybe billions of letters long. All right, well then, if I unspire, if this is just one half, then if you look over here, here is one side of my ladder, and here's the other side of my ladder. These are hydrogen bonds. So hydrogen bonds are being used to join nitrogenous bases together. The nitrogenous bases are bound to the sugars, right? And the sugar is bound to the phosphate. That we do need to understand. Nitrogenous bases are bound to the sugars. The sugars and phosphates themselves make up the sides of my DNA molecule, but the rungs are made up of the nitrogenous bases. To join two nitrogenous bases together, we have to do a hydrogen bonding. Then how we actually do this will get way more complicated later on.